And then we used only the vegetative state stress still for stem borers, but fall armyworm, they comes in the reproductive stage. Actually, these are the two contrasting phenotypes, two contrasting invert lines. And we have in that, you can see the year of 3 elite invert lines used in the breeding. They are in almost 50-60% of the hybrids released from IAMR and IARA. It is HK163, HK1105, and HK193 193-2-1, and they have been used for introgressing vitamin E and vitamin A also, genes also by IARA. And if you can see that, when we spray, you will get the complete protection. When we don't spray, HK1105 is not much affected, but the other genotypes are affected. So reproductive stress and reproductive state screening and this leaf damage screening is equally important. But so far, we have been using reproductive, this vegetative state screening only. So how the genotypes perform under multiple stresses. So we are so fortunate, we got some lines from CIMMYT, fall armyworm resistant designated lines, and they are the CML. You can see the CML 60. Those CMLs, they are having good low leaf damage. They are very good at vegetative stage, but when it comes to the seed production point of view, at the end we have to see how the company or the breeders will be accepting your line. Because every year we are releasing lines at 10% yield advantage. Also, I have seen in the Advantage presentation, in the last 11 years, from 2011 to 2023, the North India got 118 kilogram per hectare of increase, yield increase happened. The gain happened. So a breeder need a agronomically superior line. So you can see that those lines, they are not agronomically superior, but under multiple stresses, their grain yield, if you see, they are not good at foliage level, foliage vegetative state stress, but in the reproductive state stress, how much they can produce the grain. Yes, we have, but they are not on par with the giant Dr. B.M. Prasanna has shown, so on the CIMMYT lines. But yes, they are able to tolerate the multiple stresses. But one more thing you can see here is that when it is infested with actually this Kylo-1 control, this pointer is not working. So it is a Kylo, fall armyworm and control. In the case of Chylopatulus, actually, one more thing for the pathologist, when it is infested by the Chylopatulus, it is causing stalk rot also. It is an inducer of stalk rot because it is causing an injury. It can cause the entry of the pathogen. Like the insect wounding often facilitate the entry of the pathogen. That is for the fall also, stalk rot. That ear rot, it can facilitate the ear rot. So in 2024 screening again, you can see that we got the CML lines. Also, we have a line called E63. This DMR E63 is a line identified by Drs. J.C. Shekhar in 2007 and 8 before we joined. And that line is still not being used by the breeders. Why, we will see in the coming slides. Because it is a very good, it is showing resistance, but it is agronomically inferior. So we have to transfer its traits to other agronomically superior lines. Yeah. So it is in collaboration with NIPGR. It is under publication. So it is a genetic trade-off between yield and defense. So we know that this pathogen resistance, especially this not necrotrophic, the other kind, it work in the defense mechanism is activated by salicylic acid, but in the case of chewing pest, it is activated by jasmonic acid. So when the jasmonic acid pathway, when it started, your downstream genes, so many genes will be there, and in that they will, in maize, the major is benzosaxenoids. And you can see in the slide that it is our BML6 is susceptible for all. It is susceptible to stem borer, it is susceptible to TLB, it is susceptible to fall armyworm also. So, but when we tell to the breeders, yeah, it is a very good campaigner. It will give a good heterosis. So, this BML6, all the jasmonic acid and the jasmonyl isoleucine, it is the next step in the jasmonic acid pathway. It is down-regulated. But in the case of CML71 and DMR-E63, it is up-regulated. It is more in the case of DMR-63 because its basal level of jasmonic acid itself is higher. And you can see that benzosuccinoids, you can see, except the hydration product, that the product with that detoxification product, it is OH BOA. Okay. Thank you. So from the picture, the BML6, it do not have any kind of resistance mechanism to the major pest and TLB. So you can see it is a vigorous plant, and the E63 has so much of thing activated. So when you wield so many shields, your reproductive capacity will be coming low. So that's how the E63 is not used by the breeders. So we are trying to cross it with the CIMMYT lines, and in that our breeder, Dr. K.R. Yadish, he has made the E63 into CML336 crosses. No, actually it is CML33. No, it is actually CML71, and he has taken into DH facility, so he has some DH lines, and it has been screened, and he has identified. We have identified 2 resistant lines from the crosses. And it is a QTL identification through the BSA. So from this E63 and BML6, it is a resistant and susceptible line. You can see that we got 2 putative marker, and in that the breeders know better. The lot score is good only for the BNLG619. It is not enough to use these markers for screening for insect resistance, because internationally minimum 4 or 5 QTLs are reported. Based on one, we cannot. So we are going ahead with the other crosses. So it is a vegetative stage resistance to Chylopatulus. Yeah, BML6 and BML7. BML6 is very susceptible. BML7 has a very good stock resistance. So it has a ruling hybrid. It is DHM117. So in that the BML6 and BML7, how the BML6 is it is a susceptible one. When the insect attack it, it goes for the sucrose synthase gene is activated. But the BML7, it is more going into cell wall strengthening, like cellulose synthase, shikimate kinase. And you can see in that linolenic acid metabolism is activated. Linolylate 9S lipoxygenase, and it is before the JA synthesis. Then the terpene synthase is downstream to the JA pathway. The terpene synthase and terpene synthase 4. Actually, there are 2 kinds. And also we can get the terpene synthase 10. And this terpene synthase 4, one is the beta caryophyllene, Dr. B.M. Prasanna told in his presentation. So these 3 terpene synthase, you can see they are upregulated. And this is based on RT-PCR. And also after the transcriptome, we have identified, and that is based on RT-PCR. And we have gone for the metabolome also, including its hybrid. So in the metabolome, we can see that coumarin and GA, you can see that coumarin and coumaric acid. So in the morning presentation, I have seen Dr. Sundaram. He told Dr. Padma Kumari and another colleague, they made a cross between a stemborer resistant and susceptible line, and they identified QTL. And in that QTL, he has mentioned 4 coumaryl transferase. Actually, it is exactly that gene which is making coumaric acid, P-coumaric acid from coumarin. So that you can see that it is higher in the hybrid, also in the resistant parent. So what is next in the induced defense? Dr. Prasanna told we cannot, these compounds are repelling. They are not doing, Dr. Krishna Kumar, he asked a question whether it is when we antibiosis work, how it can, the human consumption, the taste for food and feed can change. Then he told no. They are repelling. So that comes in induced resistance. So we got a patent for an instrument which phenotype, which can collect the volatiles to phenotype. It is now being established in NIPSAM and IERR. NIPSAM has published some papers using the instrument. For doing this, I have again modified it. And now it is a 2025-26 ongoing research. You can see there are some resistant, also some susceptible genotype. And in that, we identified one compound, not all compounds are not there, but based on one compound, how the other compounds are performing. So in that, you can see that when in the 0R, the volatile profile is entirely different than the 24 and 48 hours. So now why IPM? So this, our breeders want agronomically superior line. They may not be resistant to pest, but we have to look for moderate resistance. So you can see that which is the yield reducing stage. See the number when the plant is at the 6 leaf stage. The number of husk is coming down. Also, the number of husk, also the grain filling, that grain yield per plant is coming down. Also, the MLB, the grain yield is highly affected by Maydis leaf blight, because when we screen in Delhi, at the end we experience high Maydis leaf blight. So it is one experiment. You can see the completely protected. These are the elite genotype germplasm of in the maize program, which has been widely utilized. At least 50-60% of the hybrid has any one of them as parent. So they are completely protected state is a T4 and zero protection is a T3. When you see the genotype HK1105, it is almost the same. There is no difference between T3 and the T4. So here comes the role of host plant resistance. But when we stage the vegetative state screening, this HK1105 is coming in moderately susceptible category, but it has a high foliage compensation. So we have to look for those kind of hybrids. And you can see the BML6. BML6 is completely susceptible. In the T4, we are getting some grains, in the other cases not. So it is a combination of insect, fall armyworm, and the MLB. So how we can deploy with IPM? So we have moved ahead with the next generation management. It is again in collaboration with NIPGR. We got the RNAi also. So we got another project with the DBT to scale up the RNAi technology. But it is, I would say, it is more of an academic. We have the botanicals also, and we have a very good endomopathogenic nematode also. And yeah, it is we developed in collaboration with IRA. We got a novel drone delivered formulation also. So in that we will put around, you need only 20 liters to spray in 1 hectare in the drone when it moves around 4 meters per second, and it has And we need around 700 gram. We'll be putting into 20 liters of water, and it is a very heavy. So there is less spray drift, and it is the latest technology. So we need to increase the growth rate 3 to 4 times to meet the E20 and E30, and IMR is into that. And so we have to balance between the moderate resistance and with plants with high vigor and grain yield, which the breeders prefer. So that's maybe the reason that the insect resistance breeding has not moved forward, because we have to balance between the vigor, grain yield, also the susceptibility, and also the multiple stresses. So IPM has a huge role. So it is how we can summarize it, pre-breeding. And the stresses we have to look for is both the stalk strength, also the ear vegetative stage resistance, also ear stage resistance. We can use a wild parent. Some of our breeders, they are crossing with the Teos Sinteo also. Then we have to deploy it with the IPM. Thank you. IMR, thank you all. And thank you for the opportunity. I learned so much in this presentation, in this 2 days session. Thank you, sir, for particularly. Thank you.
Thank you, Dr. Subi, for that overall presentation, giving the status of insect damage in the beginning, and then went on to looking at the genotypes with various stress tolerances, and ultimately leading to kind of a designated strategy to develop resistance. Thank you very much. It's open to questions from the audience. Yes, sir.
Good work.
Thank you, sir.
1 or 2 quick suggestions which I would like you to be on the. We would like to catalog the aphid species coming on maize.
Okay.
Because, and also the thrip species coming on maize.
Okay, sir.
God forbid if tomorrow maize lethal chlorosis makes an entry.
Okay.
We should have a mode of action. We cannot be totally dependent on cement lines will perform here or anything.
Yes.
So I would like normally what we do, we should anticipate an invasion and be prepared for it. So the ideal method of an invasive is we call epicenter, place around the epicenter, and then another radial. So you should develop a mechanism, and in our IIMR every year you should indicate through your coordinated cell. Anybody notices these symptoms should immediately get reported, because we all really fumbled with regard to the fall armyworm. Fault of it is also with the scientists. Nobody wanted to, everybody wanted to hide, saying that they should be the first publisher, and the moth happily kept flying 200 kilometers every day. And this is, am I right?
Sir, even though IMR, most of the ACRP parties recorded it.
You are right or wrong?
Yes, sir.
I am right on the wrong side.
Sir, you are right.
Wrong
sir.
on the right side.
Sir, yeah.
It's okay.
Right.
Jokes apart. The second species that I want you to do is thrips.
Okay.
Because thrips parvispinus and other species of thrips, they also have reported Frankliniella occidentalis. If you are not able to identify Frankliniella and different species of aphids, please get it yourself trained, so that we should be exactly in a position to tell what exactly is happening.
Okay, sir.
The second one is fall armyworm we have blown out of proportion. I am not agreeing that it is a major limitation, because one is it is only for 40 days.
No, sir.
45 days, 50 days, take it.
Sir,
okay.
it is in South India, but in North, it comes late, and you can see the ear is completely damaged and lost in some of
all
the
things said and done.
Yes, yeah.
All things said and done. The pest can be managed at
true.
this particular stage.
Yes, sir.
It's not something which is, if the rainfall is good, humidity is good, you have a very low incidence. Under dry belt, it can become very serious. But we have to give the due weightage.
Yes.
Yesterday, we made a statement that host plant resistance for insects is not ideal. more so if it is a Lepidopterous pest. When the other options are open, like Metarhizium anisopliae, you have also entomopathogenic nematodes. You can use a small oral of BT. All these things are very comfortably can be used to sustainable management of fall armyworm, because the day we discovered carbofuran, one small carbofuran granule going into the oral of the jawar or maize, we had an answer for the sorghum shoot fly.
Yeah.
So the same thing will operate even for the fall armyworm. You have a screening, but host plant resistance for insect, especially borers, has never been successful in the world anywhere. This is what we told yesterday, and you can have a genotype, but use the lesser of the options with regard to the management. But my only thing is, the management of fall armyworm in baby corn and sweet corn and other commercial crops is a different kettle of fish.
Yes.
You have large areas coming very close to between Delhi and Karnal.
Yeah, true.
Large farmers are growing baby corn and sweet
Yes,
corn,
yeah.
very close to Kurukshetra and such, that belt.
True, sir.
And I think you should really interact with those farmers and
Yes,
see
sir.
what best can be done with regard to management of fall armyworm on a crop which is economically much, much more
True,
important.
true,
Thank
sir.
you.
Thank you, sir.
Hello.
We,
Just a question. It's not a question actually.
Okay.
You have mentioned the fall armyworm and EPN.
APM.
I am from the private sector, Nirmal Seeds. So we have developed a EPN product that is Sniper.
Okay.
And it's doing well on a fall armyworm in our area.
Okay.
That's the submission actually.
Okay. Thank you, sir. I'll contact you. Oh, yeah.
Make a EPN suspension, put it into the oral.
so thank you very much, Dr. Subi.
We take this opportunity to extend our heartfelt thanks to our learned speaker, Dr. Subi Madam. I request P. Krishna sir to do the honors to the speaker. Center. Oral. Now we'll have oral presentations. We have 3 oral presentations. Sir.
Okay, the first presentation is on rapid routes to reproduction, speed breeding for maize improvement, going to be delivered by Sinchana MR.
Good evening, everyone. I'm here to present on rapid routes to reproduction, speed breeding for maize improvement. As we all know that the climatic shifts are bringing in new pests and diseases, and we need to breed for stress-resilient cultivars. And the scientists all over the world, they came up with the idea of speed breeding, as we cannot rely upon the conventional breeding approaches, as it takes longer time for breeding. And hence, the speed breeding, it is manipulating the temperature and light intensity and quality to reduce the time taken for generation of the generation interval. And but there are few disadvantages to the speed breeding. That is, there are few disadvantages for the speed breeding, as we cannot rely upon the speed breeding facilities, because it is not affordable by many institutes, as it involves investment of costly speed breeding facilities. Also, for a crop like maize, where it where it has a tall stature and dense canopy, it has limited applicability in the speed breeding chambers. Also, in the plant breeding experiments where we need to maintain large breeding populations, it is difficult and it is tedious process to maintain maintain these in the speed breeding chambers. So to address the gap between the concept and technology of speed breeding and its applicability in the field conditions, we have developed a novel technology that is natural speed breeding in maize. So in AICRP maize, we have developed this cost-effective and time-saving speed breeding strategy, wherein we can apply this technology in the natural field conditions. So this is the first report on the speed breeding in maize, which can be done in the field conditions, and it involves no additional cost other than maintaining the crop in the field. Also, we are using early harvesting as a technique for the speeding process. So the materials and methods include, we have used 7 inbreds and tested these in 49 treatments for germination percentage and seedling vigor. And we have tested first by harvesting it at 85 days. But since we did not get sufficient germination percentage at this stage, we have started from 90 days up till 120 days with an interval of 5 days. So we have a treatment set of 7 treatments. And since in the conventional breeding approaches, we store the seeds for a brief period of time before planting it in the field, we have simulated the same in the lab experiment, and we have stored the seeds of all the 7 different treatments and for a period of from 0 days to 30 days after harvest. So it makes a total of 49 treatments. So after testing all the 7 inbreds in these 49 treatments, we have crossed these inbreds and produced the hybrids, and the F1 population derived from these inbreds have been tested for the same set of 49 treatments. So the results were as follows. These are the representative photos of one of the inbred line and one hybrid line, wherein we can see that the 90 and 95 days after planting, hybrids and inbreds have shown sufficient germination percentage and also seedling vigor, which is on par with that of the control, and it provides a scope for the exploration of this novel technology in the speed breeding process. So when the graph was plotted for the germination percentage of these inbred lines, which was harvested at 90 and 95 days after planting, and it was replanted at different days after harvest, we can see that in the 0 days after harvest treatment in both 90 and 95 days after planting, it has given a reduced germination percentage of about 13%. But when these seeds were stored for 5 to 10 days, it has given a germination percentage of about 50% in case of 5 days after planting, and about 70 to 80% germination percentage in case of 10 days after planting. So it is reliable. And when the seedling vigor of GI 755 was tested, we can see that the root length and the shoot length of the 10 days after harvest treatments has given almost 9 to 11 centimeter root length and shoot length. So coming to the germination percentage of the hybrids tested at the 90 and 95 days after planting treatment and replanting at different days of harvest. So here we can see it has given a better response compared to that of the inbreds. We can see that the 0 days after harvest treatment also has given a sufficient germination percentage of about 70%. So in hybrids, we can harvest the seeds at 90 and 95 days after planting, and we can go for replanting on the same day of harvest, and we can save additional 5 days compared to that of the inbreds. When the seedling vigor of the GH21547 was tested, so it showed a root length and shoot length of about 10 to 12 centimeter, and it was appreciable. Once we have tested for both the inbreds and hybrids, we have conducted the same experiment in the next season again to confirm the results, and we have done this in 3 hybrids, that is GH21547, 150, 125 and DKC 9133, and this has given the same results as that of in the first season, that is the 90 and 95 days after planting seedlings have given sufficient germination percentage and also seedling vigor, and it was on par with that of the control. So when the germination percentage of the hybrids were harvested, which was harvested at different days after planting and replanted at 10 days after harvest was seen, the germination percentage was about 95 to 100%, and the seedling vigor was about, the shoot length was about 11 centimeter and the root length was about 8 to 9 centimeter. So this has given us positive results, and we can explore this novel technology for speed breeding in natural conditions. So once we have tested the germination percentage and seedling vigor, we have tested the root architecture of all the 3 hybrids and 2 inbreds. So within the Windriser Root Scanner 2021, and we have these are the images of the 3 hybrids, and we can see that the 90 and 95 days after planting roots has no significant difference compared to that of the control. So we can say that by using early harvesting as a technique in speed breeding in natural conditions, it has no negative impact on the root architecture of the plants. So the same results was obtained in case of the inbred lines also, and there was no negative impact on the root architecture compared to the control treatment. Since all the lab experiments that we do in the lab will be implemented on the field by the by the breeders, we have tested the same experiment in the field also. The field germination of the hybrids and inbreds, which are harvested at different days after planting and replanted at 10 days after harvest and 0 days after harvest, was seen, and the germination percentage of 0 days after harvest has given about 30%. But when these seeds were stored for a brief period of 10 days after harvest, it has given a germination percentage of 60 to 70%. And also when in the inbreds, it has given a germination percentage of 80%. So it has shown that not only in the field, but in the lab, but also in the field, we have got the positive results. These are the field germination photos of different genotypes harvested at different dates and replanted at 10 days after harvest. So, and it was seen at 8 days, the germination was seen after 8 days after planting, and sufficient germination was seen. So, there may be a fear that will there be any loss in the breeding material due to the reduced germination percentage. There might be a loss of breeding material, a slight loss of breeding material, but we can overcome this loss by maintaining large breeding population compared to the original one. The vigor of the plants at 25 days after sowing can be seen, and it can be said that due to the early harvesting, the vigor of the plants is not affected. And the morphological traits of the hybrids in the field were also tested. The ear height and the plant height were tested, and a ear height of about 45 centimeter was seen in 90 and 95 days after planting treatment, and plant height of about 145 to 146 centimeter was seen in the 90 and 95 days treatment. So there was a slight decrease in the compared to the control, but this will not affect in the rapid generation advancement or the ear bearing capacity of the plants in comparison to those plants that are grown in the speed breeding facilities. These are the GH2147 hybrids after 62 days after sowing, and across the 7 treatments, and in comparison with that of the control. These are the GH150-125 hybrids at 62 days after sowing, and these are DKC9133 hybrids. And the morphological traits of inbreds were also tested, and the ear height and the plant height of the inbreds were almost on par with that of the control, and this also has given positive results. So in conclusion, I would like to say that the speed breeding facilities has shown successful results in case of in speed breeding facilities, but it requires additional investment cost. It requires investment cost of about 3 crores, and also maintenance charges of maintaining the speed breeding facilities. But when we use the speed breeding, natural speed breeding technology, there is no additional cost apart from the cost that is required for maintaining these plants in the field. Also, when we harvest the seeds at 90 and 95 days after planting, and when we store the seeds for about 5 to 10 days, we are able to achieve a germination percentage of about 80 to 95%, and we are able to save up to 1 month in one generation, and we are able to achieve 3.6 generations per year. So this provides scope for the exploration of this novel technology in the speed breeding process for rapid generation advancement of these hybrids and inbreds. So by following this methodology, we can overcome the shortcomings of the conventional breeding approach and also the challenges with the speed breeding facilities. Thank you.
Thank you, Sinchana, for that brief presentation that you have made. Now it's open to questions from the audience. Yes, please.
It was a very nice presentation, actually. There are 2 queries, actually. What was the moisture percentage at the time of harvesting of the grains that you have harvested at 90 days and 95 days? And second one is there any significance of shank color to the early germination in the inbreds you have selected for the experiment?
Sir, we have not tested the moisture percentage, but the 90 and 95 days after harvest, genotypes will, of course, have a moisture percentage greater than that of that 100 days, other than stored the seeds. And about the shank, I have not much idea about that, sir.
Okay, thank you.
Anybody else? Okay, if there are no further questions, thank you.
Thank you, sir.
The next presentation is on genetic variability for reaction to fall armyworm among newly developed inbred lines of maize under natural condition. This will be presented by Roshni KB.
A very good evening to everyone present over here. I am Kalandar Bibi Roshni from Department of Genetics and Plant Breeding. I am here to explain our work entitled Genetic Variability for Reaction to Fall Armyworm among newly developed maize inbreed lines under natural conditions. Madhya Pradesh, Karnataka and Maharashtra being a leading producers of maize, the overall total maize production was 35.76 million tons across the India, indicates the economic importance of maize all over the nation, India. The fall armyworm, maize being a primary host for fall armyworm, it was an invasive pest. It causes an immense economic damage. Economic damages such as the leaf damage and cob damage at various stages of the maize, and it has a broad host range around 180 different species including rice, sorghum and also millets. It has a global food security threat, and also it is very difficult to control as it has a rapid migration and reproduction. Coming to the impact of fall armyworm, we can see here 30 to 70% of a leaf damage can cause the plant infestation of 20 to 80%, where we can see the plants affected, and it in turn it causes the yield loss of 15 to 50%. Under severe conditions, if we can even find 80% of yield loss. Owing to the mode of infestation of fall armyworm, an adult moth lays eggs on the underneath the maize leaf, which hatches in 2 to 3 days under warm conditions, and which turns into the young larva, which feeds on the surface of the leaf of a maize, and then it causes the various visual symptoms such as frass and window pane, and that mature larva falls into the soil and pupates for 7 to 14 days, and then it turns to the moth, which can migrate and even cause the repeated infestation from generations to generations. When we see the difference between the infestation for Kharif and Rabi season, we can see the Rabi season, the infestation is very high due to there is no heavy rainfall and also the warmer conditions for infestations, and the yield loss is very high during Rabi season. So our main objective of my study was to study the genetic variability and diversity among newly developed maize inbreed lines, which were developed from US Dharwad, that is on AICRP on maize, was to study on yield and genetic variability and diversity for yield and amylose content. During Rabi season, we found, offshoot of our experiment, we found that fall armyworm infestation under high pest pressure, we found some promising lines which was showing the various response to the fall armyworm infestation. So we have planned to record the responses during Rabi season under natural conditions with true prophylactic sprays. We can see with 5 prophylactic sprays only we can't control the pest incidence, but we have seen the promising genotypes which can be performed well only with 2 prophylactic sprays.
we have used the scale which was developed by Davis and Williams, which was later modified by CIMMYT, which consists of scores ranging 1 to 9 and responses of from highly resistant to highly susceptible with damaging symptoms. With the help of entomologist Dr. Shaila ma'am, we have recorded the damaging symptoms, and these were the plant phenotypes which were showing the frass symptoms, and some lines were also performing very well. And then we had 90 inbred lines including 5 checks, and we had a susceptible check also, CM202. In the Rabi season, we have evaluated at ACRP on maize under natural conditions. Analysis of variance, we found that the treatment significance for treatment was we found the significant difference among the treatments, indicating the variations present among the genotypes, and we found no significance between the replications, which indicates the uniformity of those lines. And looking into the distribution of pest reaction, score of maize germplasm lines against fall armyworm, we found the bell-shaped normal distribution curve with a mean score of 3.2. We found most of the genotypes showing a moderate reaction, while few lines showed lower and highest response to fall armyworm infestation, which suggests the polygenic control of these traits. Descriptive analysis of this experiment, we found that moderate variance with slightly positive skewness with leptokurtic kurtosis, which suggests that few major genes were governed rather than many genes. And we classified these lines based on the scoring developed by Davis and Williams, and then we classified our 90 genotypes ranging from 1 to 9 scores. We found that 47 inbred lines fell into the 2 to 3 damage score with the category of resistance, and we found 41 genotypes which were found to be having 4 to 5 leaf damage scores with partial susceptibility. We didn't found any highly resistant or highly susceptible, but we found lines with partial susceptible and more of resistance. This is the pie chart indicating the distribution of genotypes for fall armyworm. And we found that the promising genotypes which were resistant with a score of 2 and meanwhile they are having the grain yield of 40 quintal per hectare. The 3 lines we found GI23308, GI23206 and GI23216. So finally, I conclude that this is initial information among newly developed inbred lines, which we can further evaluate for fall armyworm resistance. And the lines which were promising, which were identified, could be further evaluated under the artificial epitophytic conditions for further confirmation and use. Thank you.
Okay, thank you for the presentation. It's open to the audience for questions. Okay, if there are no questions, thank you very much. The next presentation is Unraveling the Prevalence of Java Downy Mildew Disease of Maize in Northern Karnataka through Morphological and Molecular Identification by Dr. Prema.
Good afternoon, everyone. Respected Chairman, seniors, and all the scientific fraternity assembled over here. So I'm going to present on the topic unraveling the prevalence of Java downy mildew disease of maize in northern Karnataka through morphological and molecular identification. It's not working. Okay. So coming to the importance of maize, maize is called as a queen of cereals, and it's a C4 plant which is having wider adaptability. So coming to the major diseases which are prevalent in northern Karnataka, so it is affected by touchscum leaf blight, northern leaf blight, so curvularia leaf spot. Okay, sir. Okay, sir. So coming to different types of downy mildew, we have, so this 8 different types of downy mildew. So that is sorghum downy mildew caused by Pernosclerospora sorghum, Java downy mildew caused by Pernosclerospora maedis, Philippine downy mildew by Pernosclerospora philippiensis, sugarcane downy mildew by Pernosclerospora sakari, brown stripe downy mildew by Scleroptera raceae varieties A, Graminicola downy mildew by Sclerospora graminicola, crazy top by Scleroptera macrospora, and Rajasthan downy mildew. So coming to the morphological differences, here lies a big picture. So in case of Pernosclerospora sorghum, so the sporangia they will be oval, and in case of Pernosclerospora maedis, wherein the sporangia will be spherical or subspherical in shape, and in case of Pernosclerospora philippiensis, it will be ovoid to cylindrical, and in case of Pernosclerospora sakari, it will be elliptical, and in case of Sclerospora graminicola, so it will be papillate, and in case of Scleroptera macrospora, it will be lemon shaped, and in Scleroptera raceae varieties A, it will be oval to cylindrical, and oospores they will be almost spherical or globose in shape. So these are the certain infection characters, wherein either the oospores or sporangia they help in the as the initial source of inoculum, and most of the downy mildews they are seed borne, and they propagate or spread through the germ tubes, and lower temperature is more feasible for either the sporangia production or the germination. So this is a crazy top found in few of the germplasm lines caused by Scleroptera macrospora. So wherein the crazy top symptoms are observed, and this is a Rajasthan downy mildew caused by Pernosclerospora heteropogoni, and that one is a weed which harbors Pernosclerospora heteropogoni, and this is sorghum downy mildew, so which is due to Pernosclerospora sorghum. So these are the sporangia as well as the sporangiophores, wherein you can see the sporangia they are oval shaped, and due to outbreak of epidemic, especially in southern districts of Karnataka like Hassan, Mysore and Mandya, so our team has been formulated to survey the disease, and initially we thought that it is sorghum downy mildew which is due to Pernosclerospora sorghum. With the intention of that, so intensive survey is also taken up in the northern parts of Karnataka, and we found these kind of symptoms like downy growth. Usually downy mildew means downy growth will be there, but on the upper leaf surface also powdery growth is evident, and also half leaf symptoms are observed. Leaves become chlorotic, and stripes or white streaks, so they are also evident. And we can also see half leaf symptom and also the malformed tassel. So when we went for the survey, so an incidence of about 20 to 25% has been witnessed at Hanmanmatti, and these are the symptoms at post-flowering stage, wherein leaves turn like kedige, so that's a fragrant pine like symptoms. So and also Joyceralli and Tirumala Devarakoppa, and at Ranebennur, so all these comes under the Haveri district, and at Dharwad also the traces of downy mildew has been witnessed. And when we went for this morphological characteristics assay, we found that the sporangia, they are round or spherical in shape, and based on that, and again you can see the sporangia as well as the sporangiophores, and a Y-shaped branching or dichotomous branching of sporangiophores has been witnessed, and you can also see the sporangia which is embedded within the leaf tissue. And this is the initial formation of the sporangia, and also the zoospores and the encysted zoospores also. And for proving the pathogenicity, so we have sprayed the sporangial suspension. So by detached leaf assay, wherein the leaves have been sprayed with the sporangial suspension and kept for incubation at 80 to 20 degree. We can see the development of the mycelium as well as the sporangia at 3 days, 5 days, as well as at the 7 days after inoculation. And the reproductive structures like oogonium as well as the anthridium, so which is hiding just behind, and the ruptured sporangia which is releasing the zoospores. And usually these downy mildews, they spread by the germination of sporangia, and that's a oospore which is a resistant spore. And this is a taxonomic position of Pernosclerospora maedis, which belongs to phylum Oomycota, class Oomycetes, order Pernosporales, family Pernosporaceae, and it belongs to species Pernosclerospora, and the species is maedis. And it was first reported by Rasipovsky in the year 1897 as Pernospora maedis, and then it was misidentified as Sclerospora maedis. And in India, it was Butler who identified it. And according to that, mycelium, so it will be straight and sparsely branched, and it will be irregularly branched. And the host for this Java downy mildew are wild sugarcane, loon sorghum, corn, teosinte, Tripsacum, euclena, as well as Panicetum. And this particular disease is distributed in India, Indonesia, Israel, Japan, Thailand, and in Africa also, South America and Australia. And based on morphology, we want to confirm through molecular means also. So that's why we have gone for ITS primers as well as the LSU primers. So in case of fungi, most commonly we use ITS 1 and 4, which is a highly conserved region. But since it is a oomycete, we went for ITS 6 and 7, and also one of the housekeeping gene like LSU, which will give a clear vision with respect to the evolutionary history. So these are the different accessions taken for the comparative studies with respect to nucleotide sequence identity. And the Hanman-Matti isolate, it showed 100% identity with the Thailand isolates of Pernosclerospora maedis. And Joycery isolate also, so it showed 100% identity. And Tranebennur isolate also, it showed 100% identity with the Pernosclerospora maedis, gene bank accessions deposited in NCBI. And Tirumala Devarakopa isolate, it showed somewhat less identity of about 98.16. So based on whatever the nuclear type percent identity obtained and the sequence obtained, we constructed a concatenate phylogenetic tree by integrating both ITS as well as the LSU sequences. And we got 4 different clades, wherein Scleroptera macrospora, it formed clade 1, Pernosclerospora graminicola, it formed clade 2, and Pernosclerospora saccari philippiensis and sorghai, they fell into clade 3, and all the Pernosclerospora maedis, including the isolates taken from northern parts of Karnataka, they fell into clade 4. So based on the morphological evidences like the spherical shape of sporangia, and also the nucleotide based identity and phylogenetic analysis, we confirmed that it is Java downy mildew caused by Pernosclerospora maedis. And I acknowledge university and also our former DR, Dr. B.D. Barada sir, for giving me an opportunity. My students, Malavika and Akshata, Dr. Shalini, and my colleagues from ACRP on maize. Thank you, sir. One more thing. So these are the last slide. Last. So these are the zoospores, sir, produced by Pernosclerospora made is since students, we asked them to write a pictorial representation in the form of disease cycle wherein they write zoospores require a thin film of water and they swim for a while, then they will go for encystment. So here you can see the movement of the zoospores in water. Thank you.
Oh, thank you, Dr. Prema, for detailing the Java downy mildew disease causing organism of maize in northern Karnataka. The audience for any questions?
Sir, management in southern parts, especially Hassan, Mysore, Mandya, so there the ecosystem is different, sir. Seed treatment, it is must. There they are using F2 seeds and they are not going for seed treatment. Seed treatment with metalaxyl, that is Epron, and the seed industry. So they are also going for some changes in the polymer. So wherein the chemical sticks to the seed for longer durations so that it will have a better efficacy. And we also thought that maybe metalaxyl resistance has been developed by these oomycetes, but no, sir, it is a different species which has appeared. Sir, we have to go for intercultivation, and usually we recommend FIM. So, but the case of downy mildew is whenever the cattle has been fed by, so this downy mildew affected leaves, when it passes through the gut of the animal, so the oospores will become activated, sir. So that's one thing in increasing the intensity of the disease. And we have to go for crop rotation, deep summer plowing, seed treatment, and chemical management is essential with metalaxyl or with the Ridomil Gold, and Profiler is also working well, sir. Sir? Sir, we have other options also. Seed treatment followed by spray gives a better management, sir.
But one recommendation is cattle should not feed.
Yes, sir, because
No, in
that
a
is
lighter vein, don't take it very seriously. But what I'm wanting is, damage appears rather quite severe. And over years, it can also become even little more. And we should have, I would request to the Director of Research, whether this comes under package of practices recommendations in your university for this particular disease. If it is not there, maybe it's time to include it under package of practices. What is the best recommendation for this disease based on some of the initial field trials that they take up in their field. I think that will be a useful suggestion.
Thank you, sir. Thank you very much for the suggestion.
okay, sir, that will be taken.
Okay. Yeah, thank you very much, Prema. So that brings us to the end of the session on maize. And as you already know that there were 4 lead presentations, one each by Dr. Satish, Dr. Zarkar Subi, and Dr. Prasanna. And there were 3 paper presentations. The 4 lead presentations actually together, they gave us wholesome food on given a wholesome food on the platter to digest and probably develop strategies to enhance the productivity of maize in total. So thanks are due to the organizers for giving me an opportunity to chair the session, and thanks to the rapporteurs, Dr. Prema and Dr. Banu. Okay, I was just thanking both the organizers and the rapporteurs for conducting the session, and thanks to the organizers for giving me the opportunity, and thanks to the rapporteurs for joining me in this particular session. Thanks to the audience for their wonderful cooperation and participation in the whole events. Thank you very much.
Thank you, sir. We sincerely thank our respected chair, sir, for conducting this session very nicely. And we take this opportunity to thank him with a token of love. I request Dr. B.D. Biradar, sir, Director of Education, U.S. Dharwad, to honor our chair, Dr. P. U. Krishna, sir. Thank you, sir. Up next, we have a panel discussion. I request Dr. Surinder Tikku, sir, from ATPBR Pune to chair this panel discussion. And I request Mr. Shashi Bankar, Testing Manager, Bayer Crop Sciences, Mr. Shambhulinga Shah,